Hello and welcome everybody to my channel Bio Entrance, and today we will be seeing Central University's Common Entrance Test MSc Entrance question paper for Applied Microbiology conducted in the year 11th March 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of Applied Microbiology, you can go to my channel Bio Entrance, see the playlist. In the playlist, search for CUET and there you will find. Coming to first question, viruses they are obligate parasites. Answer is option 1. Who described microorganism as wee animalcules in his letter to Royal Society of London? It was Anthony van Leeuwenhoek. Answer is option 3. Then which of the following statement is incorrect about the bacteria? They belong to kingdom Monera. Yes. All bacterial cells are uniform in shape and size. No. There are spherical cocci, rod-shaped bacilli, spiral spirochetes, curved vibrios. So they are not uniform in shape and size. Cyanobacteria are autotrophs. Yes. Archaebacteria can survive in extremely harsh habitats. Yes, so they have asked incorrect about bacteria. So answer is option 2. Then match the following protozoa with their taxonomic groups. Entamoeba histolytica, it belongs to Sarcodina. A4. Trypanosoma, it belongs to Mastigophora, Paramecium to Coeliophora and Plasmodium to Sporozoans. So answer is option 4, A4, B3, C2 and D1. Next question, mushrooms and puffball belong to, so they belong to dash class of fungi, that is Basidiomycetes. Answer is option 2. The plaque assay is used for quantification of, that is 3, that is bacteriophages. Plaque assay is used for quantification of bacteriophages. Then match list 1 with list 2. Lichens. Lichens are good pollution indicators. They are indicators of air pollution, that is sulfur dioxide pollution. A3. Viroids are a free RNA lacking protein coat. B4. Then mycoplasmas. Then bacteriophages are protein coat called capsid, protects nucleic acid, and mycoplasma completely lacks cell wall and smallest living cell. So answer is A3, B4, C1 and D2. Answer is option 4. Which of the following is not a mutagen? EDTA is not a mutagen, it is a chelating agent. Then next one. Which of the following kinds of organisms in an ecosystem are considered as decomposers? So they are fungi. Answer is option 1. Which of the following is not used as seed inoculant for cereal crops? So seed inoculants, they introduce beneficial microorganisms to cereal crop seeds and thereby help in plant growth, health and productivity. Answer is Streptococcus. They are not used as seed inoculant for cereal crops. Then recombinant DNA technology for gene transfer has the following sequence of steps. Choose the correct answer from the options given below. So first will be isolation of DNA. 2C, then fragmentation of the DNA by restriction endonucleases, then isolation of the desired DNA fragment and its ligation into a vector, then transfer of recombinant DNA into a host and culture of host cells in a medium at large scale. So answer here is option 2, that is CABD. Then next one, which of the following is not an abiotic component of the ecosystem? Algae is not an abiotic component. Abiotic components are non-living factors of the ecosystem. Algae is a biotic component. The next one. Number of bar bodies found in XXY individual. So bar bodies, they are inactive X chromosomes and they are usually found in cells with more than one X chromosome. So XXY means one X chromosome is active and the other is inactivated So in XXY, one X chromosome will be active and the other will be inactive, forming bar body. So number of bar bodies will be 1. Then arrange the following steps in the formation of pre-initiation complex in the correct order. So TBP binds to Tata box and TF2A binds DNA upstream of the Tata box A. Then TF2B interacts with TPB. Then RNA-P2, that is RNA polymerase 2, and TF2F join the other proteins of the promoter. Then TF2E and then TF2H bind forming the pre-initiation complex. So ACDB, answer is option 1. Then next one. Which of the following interactions play an important role in protein folding? It is van der Waals forces. Answer is option 1. Then enzyme accelerate the rate of cellular reaction by lowering the activation energy. Answer is option 3. Then what are alleles? Alleles are two alternative forms of a gene. A3. Trait. Trait is an inherited character. B1. Gene. Gene, a particular segment of DNA responsible for the inheritance of a character. And recessive, an allele not expressed in the presence of dominant allele. So answer is option 3, that is A3, B1, C2 and D4. Test cross is a cross of F1 progeny with homozygous recessive individual. Answer is option 1. A color-blind person cannot differentiate between red and green. Answer is option 3. Then type of chromosome with position of the centromere we have to match. Metacentric. Metacentric chromosome has middle centromere forming two equal arms. A2. Acrocentric. Acrocentric centromere is present close to one end forming one very long and one very short arm. Submetacentric centromere is present slightly away from the middle of the chromosome forming one shorter and other longer arm. Telocentric centromere at the terminal. Chromosome has a terminal centromere. So answer is option 2, that is A2, B3, C4 and D1. Then next one. Syphilis. Syphilis is by Treponema. A4. A4 is there only in option 4. Kala Azar. Kala Azar is Leishmania, tuberculosis, Mycobacterium and diphtheria, Corynebacterium. So A4, B3, C1 and D2. Which of the following statements is true for cell membrane? It is composed mainly of lipids. A. Its fluid nature is important for cell growth, endocytosis, cell division, etc. That is also correct. Fluid mosaic model of its structure was given by Griffith. No, it was given by Singer and Nicholson in 1972. So C is not correct. It is one of the important function is transport of molecules across it. D is also correct. So A, B, D only. Answer is option 1. Psychrophilic algae responsible for imparting red coloration to snowfield is, that is by Chlamydomonas nivalis. Answer is option 1. Crossing over occurs during the Pacatine stage of prophase 1 of meiosis. The sequence of phases in the cell cycle is, so first will be the G1 phase, then S phase, then G2 phase and then M phase. So B, D, C, A, that is option 2. The lag phase in a bacterial growth curve does not occur under the following condition. When inoculum is taken from an old culture, you have lag phase. They have asked does not occur. When inoculum is taken from frozen culture, again lag phase is present. When optimum temperature and pH conditions are provided, yes. When inoculum is from exponentially growing cells growing under similar conditions, you don't get a lag phase. They are already in the exponential growing phase. So answer is option 4.